+2 Biology, the chapter Human Health and Diseases, and our topic of discussion today is the disease cancer. And you know, cancer is the most dreaded disease in the world. Millions of people died because of cancer. Again, millions of people suffer cancer today. Why cancer is such a dreaded disease and what happens in cancer? So let's go to the details of the disease cancer. So dear students, when we discuss about the disease cancer, we have to discuss some major areas like what is oncogenic transformation, means what happens during cancer, and what are the agents of cancer, or which are the carcinogens, detection and treatment of cancer too. It's given in a very brief in your textbook, and any elaboration you can do with extra reference, and we also discuss some more matters in the live session. So when we discuss about cancer, okay, you know our body cells, the growth, the reproduction, cell division, its development, differentiation, all the features of the cell, each and every cell of our body is coordinated with number of proteins. In each stage of cell cycle even, there are specific enzymes functioning. Such a coordinated action our cell division is. Okay, that means in a normal person, in our normal body organs, the cell division is a coordinated activity. Whereas, when a normal cell becomes cancerous, what happens in cancer? Okay, a cancerous cell, first and most important thing, when a normal cell is undergoing oncogenic transformation, cancerous transformation, we can say in our normal cells, there are proto-oncogenes and cellular oncogenes present. I mean, within our cells, within everybody's cells, there are certain genes called P-onc or C-onc. Proto-oncogenes, proto-onc or C-onc, cellular oncogenes. What are those genes? These are the genes which can be transformed to oncogenic, which can be transformed to carcinogenic under certain situations. That means these genes, when activated, it leads to cancer. So our normal cells contain proto-oncogenes and cellular oncogenes like genes which can be activated to cancer or oncogene under certain conditions. And all such sort of condition, what can change this C-onc to oncogene or P-onc to oncogene? All such stimulants, all such agents we can call as carcinogens. Okay, and presently there are thousands of carcinogens found. Okay, even what we eat, what we expose may lead to cancer. Okay, there are so many radiations, chemicals, okay, even biological organisms which can act as these agents of bringing cancer. Okay, it can be due to simple DNA mutation, due to any of the factors, or due to some external viruses or anything. We are moving to that. Anyway, understand the first session. Proto-oncogenes or cellular oncogenes with the help of what carcinogens can be stimulated to oncogenes which can lead to cancer. Okay, this is what we call oncogenic transformation. Cancerous transformation. Then, when we discuss about the normal cell, what is a normal cell? Okay, in a normal cell, it is in a form of group of cells. It is inside a tissue. Okay, its growth is inhibited because of the neighboring cells. I mean, a normal cell is unable to grow and multiply by its own to a maximum extent because it shows contact inhibition. Whereas a cancerous cell does not show contact inhibition. So there is no any inhibition due to contact by neighboring cells. It can expand and it can divide its own way. Okay, so in a cancerous cell we can say it loses or it has lost the contact inhibition. So it grow uncontrolled and divide, shows rapid proliferation and is leading to a growth known as tumor. Okay, so tumorous growth of cancer is due to the result of uncontrolled growth and cell division, proliferation of the cells. Okay, and see, sometimes other type of tumors may be there in our body. Some of these tumors may not be neoplastic. If it's a cancerous tumor, we can call it as neoplastic tumor. The term is related to what? Cancer. So neoplastic tumor consists of neoplastic cells. This is cancerous tumors. There are some other tumors in the body may not be cancers. Non-cancerous tumors are also there are also possible in our body. So anyway, when we discuss about the tumors, keep the two terms in mind. What is a benign tumor and what is a malignant tumor? Okay, are you familiar? Malignancy, the word you are familiar. Okay, so benign tumor is a tumor which will remain confined to the original location. Where it is initially found, there only it confined to. That means it cannot spread to other areas, to the other organs. So keep in mind, benign tumors are the tumors which do not spread to other organs or other areas. Okay, it will remain confined to the original location. Whereas, what are malignant tumors? Same way, okay, malignancy is a dreaded property of cancer, you know. Okay, so malignant tumor means a tumor which can migrate or which can invade into other organs, which is spreading type. Okay, a tumor which does not remain in the original location. Instead, it can spread to other areas, other organs. Is called what? Malignant tumor. So we can say malignant tumor is invasive in nature. It shows invasion to other organs. See, some of the tumor cells, malignant tumor cells, if it's sloughed off, okay, if it is sloughed off from the original tumor, it may reach the blood. Okay, through blood, it can be transported even to very distant organs. Okay, so almost different parts of the body can be infected. You know, if it's different stages of cancer, you are familiar. First stage, second stage, third stage, fourth stage. Like when the disease proceeds, you know, it's very difficult to control it. And if it's very early detected, you know, it's very easy to control nowadays. Okay, so anyway, what is the fourth stage, third stage cancer? It means what? It has spread to the other organs too, through blood. Okay, just even if it's present in the cancer present in the bone, okay, bone marrow, it may be reaching to the different portions of the body, different parts of the body. Maybe in the brain and it may be in the heart, and almost all organs will be affected due to this invasive property of the malignant tumor. So malignancy is a dreaded property, uncontrolled division and forming tumor, and this malignancy means it can spread to other cells. Malignant tumor cells can be sloughed off and it can spread to other organs or other parts of the body. And this invasive property, and this is known as metastasis. This invasive property of cancer is known as the most dreaded property of cancer is known as metastasis. Okay, I think it's clear. These are some simple topics. Okay, then let's discuss the other side. The agents of cancer. What are the agents which cause cancer? This is an area where most of the research works are going on now. Okay, what leads to cancer? How can we control this? Anyway, carcinogens are the name given to the agents of cancer. Cancer causing agents are called carcinogens. These carcinogens can be either physical or chemical or biological carcinogens. So familiar examples only given here. Okay, physical carcinogens means the radiation. You know that the radiation, it can be ionizing radiation, non-ionizing like UV radiation, ionizing radiations like alpha, then X-ray, gamma ray. This all people working in such field. They all are susceptible to this type of cancer. This type of cancer. So, physical carcinogens include radiations. Then, chemical carcinogens, you have heard it recently, Ajinomoto, okay, nicotine, the tobacco smoke is leading to what? Smoking is leading to lung cancer, the oral intake is leading to mouth cancer. In theatre and media, you are familiar this advertisement. Okay, nicotine is a carcinogenic chemical. There are a number of chemicals found to be carcinogenic, which can convert the cellular oncogenes to oncogenes. Okay. Anyway, the chemical carcinogens, one example is nicotine. Then other one, biological carcinogen. Biological carcinogens are biological agents. Means they are viruses. These viruses can cause cancer. These viruses contain viral oncogenes. So they can bring cancer to us. Okay. So these are the major agents of cancer. The agents of cancer are called carcinogens. They are physical, chemical and biological. Physical, the radiations, chemicals like nicotine. Biological organisms like viruses, oncoviruses or oncogenic viruses. Okay. Then let us discuss what are the detection or diagnostic techniques of cancer. Okay. The diagnosis has a crucial role in cancer. Okay. Early detection of cancer is giving complete cure. Okay, if it's detected in a third or fourth stage only, it's very difficult to control. So, early detection of cancer is the most significant area to discuss. It is possible nowadays by a number of techniques like ELISA, antigen-antibody interaction, PCR, okay, then DNA hybridization, RNA technology. So, there are so many recent techniques to detect cancer early. Okay, anyway, the cancer detection techniques, we can just have a brief sketch. See, listen, if it's blood cancer or like, we can very easily detect it by blood test or bone marrow test. So, blood test and bone marrow test is one method of detecting cancer. It depends on where the cancer's growth is or where it is. Okay, anyway, the blood or bone marrow test, then biopsy and histopathological study. And mostly the cancer produce some sort of tumors, and this tumor is growth, some cells can be taken and can be observed under pathological lab. Okay, this is what is called histopathological studies. Okay, usually if there is tumor is growth, that piece of tissue will be sliced into thin sections and it will be observed under microscope. That's what the technique is, biopsy. In biopsy, the suspected tissue is sliced into thin sections and it is observed under microscope after staining. Okay. In any way, biopsy is a very popular term nowadays because of this cancer, and this any part of the body, any part of the suspected tissue is given for biopsy. And even if it's any other tumor or any other growth in the body, that we are sending it to for biopsy now to check whether it's cancerous or not. Okay, so biopsy, histopathological study, then internal organs cancer. How we detect it? Usually you are familiar, endoscopy to locate the growth. Okay, anyway, endoscopy is there, and the detection techniques include internal organs cancerous growth. Detection technique include X-ray radiography, CT scanning, then MRI scanning, PET scanning, PET scanning, etc. What do you mean by the scanning techniques and X-ray radiography? In X-ray radiography, X-rays are passed to the body and we can get the images in a sheet. Okay, you are familiar. This X-ray radiography uses X-rays. Okay, and we get a 2D image. Whereas same X-rays are used in CT scanning. CT scanning also uses X-rays. But we get a three-dimensional view because it is computerized. CT, computed tomography. Its expansion is computed tomography. It uses X-rays, but we get a 3D image with the help of some computerized techniques. Then PET, positron emission tomography. Here, the technique is based on the emission of positrons. Okay, anyway, it will be different in normal cells and tumorous cells. Positron emission tomography. It will correctly locate and correctly sketch the tumor size and intensity. PET scanning. It's popular as PET scanning. Positron emission tomography. Computed tomography. Then MRI scanning is another most common thing. Magnetic resonance imaging. Magnetic resonance imaging. Here the person is introduced to a very strong magnetic field, and this is sketching or this is analyzing the magnetic properties of water in the body. It differs in the tumor and in the normal cells, so it can clearly sketch the intensity or the tumorous growth. So MRI is magnetic resonance imaging. Here there is no harmful radiations used, that's the advantage, and it depends on the magnetic property of water in a person's body, which is different in tumor and the normal growth, normal cells. Okay, that's what is MRI scanning. These are all in very brief given to you. Understand, CT stands for computed tomography, it uses X-rays. PET stands for positron emission tomography. It depends on the emission of positrons. The person will be given a glucose drink activated, then it will be wherever the glucose reach, it releases the positron, so it is sketched. Okay, then MRI scanning, magnetic resonance imaging. It is using what? The strong magnetic field in which the difference in the water magnetic property is giving the tumorous growth identification. Anyway, the recent techniques we have already discussed in the biotechnology chapter: antigen-antibody interaction, PCR, ELISA. Okay, antigen-antibody interaction, the advanced version is ELISA. Then DNA hybridization technique, PCR, all such techniques are used. Okay, here we can just, generally you can just mention as rDNA technology. rDNA technologies are recently very helping in early detection of cancerous growth. Okay, that's what is about the detection of cancer. It's very important in the control of cancer. Early detection is significant, having significant role in cancer treatment. Then about the cancer treatment. All of you are familiar with the word chemo related to cancer. Then which are the common treatment strategies of cancer? Cancer can be treated by the 4 major techniques: surgery, radiotherapy, chemotherapy and immunotherapy. Okay, then what do you mean by surgery? The cancerous tumor is surgically removed. Okay, there are some problems. There may be some cells left even. That itself may lead to next spreading. Okay, anyway, surgery, surgical removal can be a better option in early stages of cancer. The surgical removal of tumor, surgery. Then the second method, radiotherapy. It is a method which requires utmost care. Radiations are given to the cancerous cells to irradiate it. It is completely killing the cancerous cells, but it should be with extreme care not to destroy the normal cells. Okay, this is a problem faced in radiotherapy. Radiations, using radiations, cancerous cells are irradiated. That is what this radiotherapy is. Then chemotherapy. What is meant by chemotherapy? Using chemicals. Okay, chemicals which can shrink the cancerous growth, or it depends on different cancers, what type of chemicals used, and also it depends on the person's health. The chemotherapy is. Anyway, the chemotherapy using chemicals, the cancerous cells are controlled. Okay, the person is given with the chemicals. Okay, then immunotherapy is the introduction of certain immune system modifiers to the person. Okay, that is usually alpha interferons like chemicals are introduced, and it can just supplement only. Anyway, so a combination of these techniques are usually used: surgery, chemotherapy, immunotherapy, a combination of these 4 techniques, any 3 or any 2 is usually advised by the doctors depending on the severity of the disease. Okay, so the cancer treatment, 4 approaches: surgery, radiotherapy, chemotherapy and immunotherapy. Immunotherapy, mainly immune system modifiers like alpha interferons are introduced. alpha interferons. Keep in mind, you have already studied what are interferons. These are the chemicals which spread virus infection to neighboring cells. Okay, anyway, in immunotherapy, alpha interferons are used as immune system enhancers or modifiers. That's about the treatment of cancer. So in your textbook only this much is given about the cancer to study in connection with plus 2 syllabus, but you can have an advanced reference learning, and also we can discuss all the doubts in the live session. Thank you.